So thanks very much, and as Mike mentioned, there was, there we are. Do I advance this, or do you? Thank you so very much. So as Hal mentioned, there was actually a conference in Northern Virginia, and Eli Lilly contributed with it, and it was a sort of getting to know the potential for industry-academic collaboration in Virginia. And so this really does kind of represent a follow-through, and to some of the remarks that were made at the beginning, we know precision neuroscience is a reality. What are we going to do about it? So I've tailored my presentation today to describe how industry is approaching things. How are we doing something about it? And just in reference to the background, I'll fill it out even a bit more. My high school was T.C. Williams in Northern Virginia. Remember the Titans? The college was in Williamsburg. And then medical training, the MD and PhD were in Richmond, and then Charlottesville was my first faculty position in my medical residency training. So I've been coming here I've been trying to get here. I've been working my way west on 64. Now that this medical center and this conference exists, I've come home. I did actually teach for 1 week at the Virginia College of Osteopathic Medicine, but it's a pleasure to be here. And it's a pleasure to try to connect the dots, especially between the first two talks we've had today, where Susan demonstrated biological understandings that are important, and then Jonathan talked about heterogeneities that could be detected. That's the role of industry, is to take these understandings and from them to bring forward chemical entities, but also to bring forward clinically relevant detectors of effect and potentially the patients who should or should not receive these chemical entities. And so it is a collaboration between academics and industry that will in the end produce the best drugs for the right patients. And this is what we call precision medicine, and that's my area of Eli Lilly, is precision medicine and neuroscience. I will just present for you a recent publication which codified everything that we sort of knew. Pharma is a willing recipient for precision medicine and for biomarkers in clinical trials based on disease understanding and the identification of heterogeneity. And this group asked, over the last 15 years, what is the likelihood of success going from phase 1, phase 2, or even phase 3 into approval with or without the presence of a patient selection biomarker? And that's one kind of biomarker. And the important statistic is on the far right. You have a 3 to 4 times greater likelihood of registering a drug, which means putting a therapy in the hands of patients and physicians, if you have been using a precision medicine biomarker in your trials. So this effect is not lost on pharma, but it had not actually been presented in this way before. So we are a willing recipient of this information. This is the value statement that I put out to pharma, the internal lobbying that I do to get funding for the initiatives that we undertake. I am with Eli Lilly now. I was at the University of Virginia for 12 years and then in Texas for 4. So I've been an academic physician scientist for most of my career. I've studied, in fact, Parkinson's disease, mitochondrial dysfunction, and Alzheimer's disease as particular areas of focus. So I come to pharma, I've only been there about 3 years, so I am sort of a stranger in a strange land, but I'm beginning to figure out the sort of things that they care about. It's not that they're different than academics, it's just that they're orthogonal. So I've come to conclude that academics are rewarded for building an expertise and then continuing to build on it in a very vertical kind of a way. And so if you go to Carl Svoboda's lab, you go because of what he has done, not what he's doing tomorrow. And if he decides to do something completely different, that's probably not a good thing. It's probably not a good thing if a cardiothoracic surgeon decides to become a dermato surgeon. The residents that come there come for their cardiothoracic training. And so academic excellence is rewarded for staking out an area and then continuing to build expertise and knowledge in that area. But it's not really very transitive. Pharma is the entire opposite. Pharma is required to move. If you stay in any area for longer than 20 years, you outlive the patent. And so even over longer courses of time, pharma is required to move from one area to another according to its ability to innovate in that area. They're not allowed to stay there for very long. When pharma and academics intersect most profitably, it is as if pharma is driving through the countryside and Roanoke represents academics, and for one brief shining moment, they're in the same area at the same time. That's when the collaborations can be most productive. because pharma has to deliver on a timeline. And so where I find myself caught between a rock and a hard place is when I see something coming that's of obvious value, but I know that I won't be able to bring it into a study in the same timeline as the studies move toward registration. And that timeline can't be disturbed in pharma. And so that represents much of the difficulty that academics has in interacting with pharma, is simply that their timelines are very unforgiving. And so they pass by and keep going before there can be reasonable traction. And I'll discuss a little bit of that. But having said that, though, Eli Lilly is among the top as far as the utilization of these biomarkers. So we are positively disposed toward precision medicine and the biomarkers that enable it. Just a little bit of background on what I call things biomarkers. I mean, they're indications of a physiological state, condition, or response. They can be qualitative, so my group has a genetics, genomics, pharmacogenomics component with pain, psychiatry, and neurodegenerative disease represented equally. There are quantitative biomarkers. These are the ones that are typically most familiar to physicians who order tests. So these are protein-based very often. Don't have to be. They can be measures of amino acid degradation, for example. But these are quantitative statements of the amount of some protein or other biological entity. It can also be imaging studies. So Lilly has developed imaging agents that reflect amyloid and tau deposition in the brain and can quantitatively state where and how much of this is there. And then there are functional readouts, and I'm going to spotlight that today because I think that's one of the most promising areas of precision medicine implementation. And these are what I refer to, or what the field is sort of referring to by the nickname of digital biomarkers. That's a digital biomarker. So if any of you have welcomed a smartphone into your life, you've experienced the way that it works its way into much of what you do and has transformed much of what you do. I'm not sure most folks would return to regular banking having gotten used to online banking. These aren't in clinical trials to any great extent, but it seems quite obvious to us that if their potential can be realized, then we can disrupt the clinical trial in a very positive way. Just as Googles and Androids and such do, we can mine the data of the patients in the trial to identify heterogeneities. And if we can understand the genetic or disease understanding basis of these heterogeneities, then we can make advances very quickly. We can get data during the trial. So I'll spotlight that a little bit later. I'll tell a story of our first foray into that. Generally speaking, there are two different ways pharma will develop biomarkers, in this case for precision medicine. And my simple rule is if the biomarker helps identify different disease subtypes, for example. So if we were funding Jonathan Flint, for example, the best move isn't for Lilly to undertake that specifically. The best move is for all interested pharma active in psychiatric disease and depression in particular to band together and in a consortium fund him and investigators like him to have a comprehensive approach toward developing this understanding of disease. This is the rising tide lifting all boats. And pharma will benefit from this understanding, both in terms of initially identifying targets, and then when they get to registration at the end, the registration authorities will recognize this categorization of disease because it's been independently undertaken by academics, not funded by any particular pharma. The downside is that this tends to take longer, so it develops the field rather than the therapeutic in particular. Internal development is best when you're, instead of looking at categories of disease, you're looking for categories of response. And so Pfizer or Merck are unlikely to be interested in funding a test that detects increased responsiveness to a Lilly drug. This is Lilly's job. And so we can move very quickly, we can fund all elements of the work and the collaboration, but the flip side of that is that it's pinned to a particular timeline, and what we're looking at is response to a Lilly therapeutic, which may not be the primary interest of an academic investigator who's developing more general knowledge. The path to development, I'm not sure I need to spend too much time on that, but basically, we can build it ourselves, and if we do, we have more speed and control, but it takes more capacity and expertise, so I have to lobby for personnel, capabilities, capital to do that, and pharma moves in one-year cycles typically. I can work externally, and I can either do that by finding a biomarker that's already there and bringing it in. That helps if I'm trying to meet clinical trial deadlines. But it presupposes that one's already there. So we have either fertilized the field through consortium funding, having good foresight and having done that several years ago, and these are now ready, or we just take our chances and try to understand whether they're there. And the external consortium helps to develop the academic understanding of disease and move it forward so that these are available once clinical trials begin to emerge. And they each have their pluses and minuses, but those are our approaches. Now I'm going to pull digital biomarkers out in particular because, especially the comments in the beginning, what are we going to do about it? So we know these things are out there, and we know in the end someone's going to get it right and they're going to turn clinical trials upside down. Hopefully for the better, but it might be a bit of a bumpy ride in the beginning. And we don't really know what we're doing here. And so we have undertaken the implementation of one of these in a trial, and I'll describe that. But let me just describe in general the approach to digital or any other biomarker. As I've said, you have to start with the disease understanding. The last thing you want to do is have the cell phone start sending all sorts of data to you, and you have no idea how it correlates with the disease. Because you will find trends and you will find things, but what you don't know is what those things are. You might find some very interesting changes with drug, but is the patient dropping the phone and his kid's playing with it? And so you're suddenly getting all these changes, or is the patient having a drug effect? So there are understandings that you ought to have, and then you ought to, as any physician would counsel, only order the test that will help you make a decision. So you ought to understand the disease, and then you ought to only make use of the data gathering ability of these, not only the phones, but the wearables, the Fitbits, and all of the other coupled devices that can bring in this kind of information. You ought to only monitor the things that you understand. And even then, you're going to have to make sure that you're seeing what you think you're seeing. So disease understanding is critical, and when we evaluate biomarkers, but these digital biomarkers in particular, we ask, based on our understanding of disease, is this measuring what we wish to measure and nothing else? We target functional domains. This is a functional biomarker, so the patient interacts with it. And so they can interact with it many times a day, or if it's a Fitbit or a cardio monitor or something, they can interact continuously with it. It can be active, so for cognitive tests, depression, mood, psychosis, anxiety. They can interact with various games or iPhone-enabled presentations, and that interaction can change according with their mood, and so we can monitor them daily, we can monitor them monthly, weekly, hourly. And we can do it in relation to their taking of the drug and during periods before and after the trial when they're not on drug, so we can determine whether there is a drug effect. There are a lot of passive bits of information that are gathered from an iPhone as well. So when you interact with your phone, when you start tapping it, there are something like 52 different independent data points that are gathered based on whether you're tapping it in the same position, what your frequency is, your coordinate when you tap it twice, how coordinated those are, what the rhythm between the two is. So there is a lot of passive information just from touching the face of the phone. And this passive information can be brought in along with the active information that you're assessing. You can use these to try to identify patients with disease versus patients without. And then the second case is, generally that's a phase 1 study where we would bring the digital biomarker in and see if it can recognize the disease patients if they're there. The phase 2 study is, do the disease patients respond to therapy? We have the opportunity to bring the discriminating signals forward from phase 1. We ask many questions in phase 1 from these digital devices. Those signals that seem to be best at disease versus non-disease come forward into phase 2. Then we ask, can we see an effect with drug? If you see an effect with drug, those signals come forward potentially into phase 3, where they can be locked down and you can actually begin to approach a diagnostic or theranostic device, where it might be part of the phase 3 study and then might eventually be part of clinical practice, where a patient might use this app to determine whether they're a candidate for drug, or within 1 or 2 weeks on drug, whether it's having the correct effect. And so in the end, you're building something to enable precision medicine in the clinic. So let me discuss an example, and it ties to the first 2 presentations you've heard today. So Parkinson's disease is the target of the mPower app. And this app was constructed to measure 3 domains of Parkinson's disease. along with cognition. And so Lilly has Parkinson's disease drug therapeutic trials underway, and so I'll talk about one of those. The mPower app was developed by Sage Bionetworks. According to the criteria that I've used, do we have a disease understanding? We do. We have a pretty good disease understanding of Parkinson's. The neuropathology suggests that it, at least in the canonical sense, by Brock staging, it progresses up from the fourth ventricle into the midbrain. The locus ceruleus is affected early, and then you have the appearance of clinical symptoms when you lose more than 60 to 80% of the neurons in substantia nigra. You interfere with the palatal signaling, and then you may get progression into the cortex where you begin to get Lewy body dementia in association with Parkinson's disease. And so we can measure domains that track this disease. This had the added benefit that they've already had it out in the world for over a year. There have been 50,000 downloads. In that sense, it is the largest Parkinson trial ever. Patients download it freely. They identify themselves as patients are healthy, and they self-report on their taking of drug. And so they had a large database of discriminating disease versus control, which was useful to us in evaluating it. And also the patients would take DOPA, and so they were able to do some pulling forward of response to drug in the disease discriminating signals. And this was also useful to us because we could start at that point, which is exactly where we needed to be to bring it into a phase 1 trial. So the functions that are assayed as you move up from the brainstem is voice first, because the locus ceruleus has the recurrent laryngeal, which innervates the larynx and the pharynx. And so if you have failure of dorsal motor nucleus of vagus. So if you have failure, and nucleus ambiguous, if you have failure in those neurons, mainly because they're dead and they're not there anymore, then you have the inability to hold the shape of the larynx for an extended period of time. So the patient goes, ah, into the phone. And you either have a strong, brisk waveform, which is healthy, or you have fluctuations, because they just don't have the innervation that's needed to maintain the shape of the larynx. And those who have interacted with patients with Parkinson's will be able to say that there is a change in the tone of their vocal caliber. As the disease progresses forward through the locus and then eventually into the substantia nigra, then you're going to start to have motor deficits, and this is generally the clinical appreciation of disease. There's heterogeneity here. It may be in the arm, it may be in the leg. It's not always one or the other, and it's not always synchronous. In fact, it's usually asynchronous at the beginning. So you assess the arm through tapping, and so they do a rhythmic tap with both hands for about 10 seconds. Now you're counting the number of taps, but you're also doing all this passive data. How regular is their rhythm? Are they hitting the same spot? Or do they have this resting tremor that causes them to bounce all over the phone? So you pull in a lot of data. And then you also assess gait and balance, so they walk and they stand. So you're really doing a UPDRS-like motor examination of the patient via the iPhone. And then you assess their memory through a spatial cognition where different florets light up at different signals. And so you assess that speed. I'm going to move a little bit more quickly now through things because really I'm at what I meant to say. This is how one takes a new technology, a new biomarker, and tries to bring it into a trial. We try to understand the disease, and then we bring it to the patients in the trial and look for drug effects that are rational to us. And then we can use it for other purposes. This has been referred to as N=1 trials. So the patient becomes their own trial. So I'll give you some of the insights. One of the patients that we see, they have in the tapping, it's not necessarily the number, but it's the rhythm between the taps. So it's some of this passive data. The standard deviation is quite large. So they start, they stop, they start, they stop. As soon as they go on drug, this tailors down very quickly into a very regular rhythm. This is the same sort of thing to patients coming on dope, but regaining motor control, regaining rhythmic potentials. So we can see that in numerous measures that are passive, but the interval, the rhythmic tapping is the one that's sort of the briskest and might be carried forward into a companion diagnostic to determine drug effect. And when the drug is removed, then you see a return to the baseline almost immediately. It's a very powerful signal. Now, another patient doesn't show this signal. However, their tapping is very regular to begin with. And so as you look through some of the other domains, you come to the conclusion that they probably don't have a manifestation in their arms. What you do see is a manifestation in the walking, in the gait. So really, you have to do a summative physical examination. Each patient is different. They have to, in a sense, be their own control. The way we organize this trial, there are 2 weeks before intervention, 2 weeks of intervention, 2 weeks after intervention. And what you look is to establish their baseline, and then the statistics compare them against themselves. When you're doing near continuous data, you can get enough data points where the patient becomes significant or non-significant. Then the trial becomes a meta-analysis, really, of the trial participants. This is very challenging. The discussions I've been having the last couple of weeks have been through the statistics and the advanced analytics group, who are not capabilized to do this in a formal way. You will find that pharma is the most conservative group you'll ever encounter, and this is something new. I'll tell you that in bringing this into trial use, it's not inconsequential. You've got to come to an agreement with the external source. You've got to bring it in. There are many cycles that have to be underdone, but eventually you can get it in and you can begin to get data. Implementation is as I've described. Our learnings have been start early. It's a new approach for many areas, so you've got to kind of fertilize the organization to understand that this is coming. Start exploratory, don't bet the trial on it. Iterate often. We have 20 updates of this app, 17 of which were in the first 3 patients. Be able to push them onto these phones. And you have to, right now we're dealing with the fact that when I look at one of these, and I can look at these every day, I see the patients, they do these 4 times a day, I am unblinded, meaning I can see drug effect. That's a big deal, especially if it's a blinded study. So this is another thing that I'm having to push against in pharma. Basically, I'm pushing against walls that were set up for something different than precision neuroscience in real time. It's a paradigm shift, and right now, we're at the beginning. Basically, everything can go wrong, and I can talk more about each of these points. I'll show them to you now in the discussion. It does enable rapid decision-making that should reduce our size, time, and cost, but you have to do that the right way because in the end, it has to pass regulatory muster, and this is all very new. It is an entirely different trial design, potentially adaptive, but adaptive trial design has a lot around it, and so the fact that we might be able to do it doesn't mean we can do it. You could even see your way in the real world toward adaptive dosing, where a patient's app might monitor their response, and the pharmacy might not distribute 50 milligrams, but 25.8, and then 32.5, based on the app bringing them down to no response and then bringing them up to response. So if you have a safety signal higher, you might have different dosages personalized for each patient. And as neurons continue to die with disease, you might have the continuous upgrading of the dosage precisely tailored to the appearance of the pharmacodynamic signal. And this could work in real time and be specific for every patient. And that's the potential future, a real-time, patient-specific precision diagnostic to tailor every drug precisely to the patients who respond and at precisely the right level. I'll leave it at that. I'll acknowledge Stephen Friend and Sage. Hal's been great getting everything set up for this conference. Lily's Clinical Neuroscience has been a spectacular group of people. It was their quality that convinced me that that's a place that I ought to go. That many right people can't be wrong, and especially the patients who participate in these trials. Thanks, and I apologize for any overtime.